Muchas gracias a los organizadores de este Congreso de Aerognología. Es un verdadero honor para mí estar con ustedes hoy para hablarles de trabajos que hemos venido realizando con mis alumnos desde hace bastantes años en la Universidad de British Columbia y anteriormente también en la Universidad de Arizona. Y quisiera, bueno, como ven por el título de mi charla, decidí por varias razones dar la charla en inglés. Espero que haya sido una buena decisión, pero por favor siéntanse libres de hacerme preguntas al final, ya sea en castellano o en portuñol. Y quisiera empezar con, con esta cita de Michelangelo. Every block of stone has a statue inside it, and it is the task of the sculptor to discover it. And I feel that we as scientists, biologists, and in our case, our ethnologists, our task is to observe the organisms that guide us with open eyes and open minds as they reveal to us the secrets of how nature works. And this is what I have done in my work, in which I have had the fortune to encounter these extraordinary organisms, the social spiders. And here's a sample of these spiders. I will be telling you a bit more about them. But in the quest of understanding various aspects of the biology of these organisms, I have explored fascinating areas in ecology and evolution, including sex ratio evolution in metapopulations, multilevel selection, cooperation and nonlinear dynamics, inbreeding and its consequences, and more recently, the ecology of social evolution and sociality and community assembly. And it is on these two areas that today's talk is going to be touching on. And these two areas relate to the field of community ecology, which basically asks the fundamental question of what determines the composition of plant and animal communities. And there are two types of processes, mechanisms involved. One is referred to as an environmental filter, which basically suggests that organisms occur in different areas depending on the match between their organismal traits with specific environmental conditions. So here in this example, for instance, we see a range of habitats from dry to moist, and we see that the species here shown in blue do very poorly in the dry areas, but they do well in the wet areas. And the opposite is the case for these species shown in orange. And what I'd like to explore today is how this environmental filter may relate to understanding why spiders are social in some areas and not others. The other process that is expected to be involved in determining what species are found in particular areas is referred to as niche partitioning and limiting similarity. And the idea here is that the species that occur in an area need to be sufficiently different so that they will be able to coexist. Otherwise, one species would outcompete the other. And I will be talking a bit more in detail about this, but one of the messages that I like to give you with respect to this is that sociality and group living may play a role in structuring ecological communities. So my talk will consist of three parts: a brief description of the social spiders and the fascinating biology. Then I will address the question of what may be the environmental filters of spider sociality and how sociality may play a role in niche partitioning and limiting similarity. And the second question is more or less addressing the second point is addressing the question of why and where social spiders are social. And the third one, how does sociality change the spider's ecological niche? As you know, the vast majority of spiders are solitary. And here we just have a photograph of the nest of a solitary species. Basically here, offspring are close from the egg sac, but they disperse soon thereafter. And they lead pretty much their entire lives as solitary individuals. But in a few cases, several genera actually in different spider families, rather than dispersing, the offspring remain together for a period of time, often overlapping with the mother. And during this time, they cooperate in maintaining their web, they cooperate in prey-capture, they share their food, but then they disperse before reaching reproductive maturity. We refer to these species as subsocial, and their nests contain at the most a few dozen spiders, typically the offspring of a single female. But in the species that we refer to as social, rather than dispersing, the spiders stay together in these nests. They mate with each other after they mature, And they produce offspring that, again, reoccupy the nest and may enlarge it. And these colonies may grow over multiple generations to contain hundreds, thousands, or even tens of thousands of spiders. And this is going to depend on the species and also where the species is located. All these different species that I'm showing you here, photographs of their nests, belong to the genus Anelosimus, the genus in the family Theridiidae. But this is not the only genus where the social species have originated. In fact, there have been there are about 19 species with this sort of the higher level of sociality that I described, the social species, and they are distributed around the spider phylogeny. So this is a tree of all spiders, showing here the number of independent origins of this form of social behavior. And we see the genus Anelosimus here in the lower part of the tree is one of the 3 genera where social species have originated. And interestingly, practically every species in that genus, and for that matter, in every genera, seem to be an independent origin of sociality. And that is an interesting question as to what that may be. I won't have time to talk about that today, but we could maybe talk about this in the question period. So as I say, social spiders are phylogenetically diverse. Here is the nest of species in the genus Anelosimus. Another species in the same family, but a different genus, Theridiidae nigro-annulatum, and this is a nest here that would be the nest containing a single female, or perhaps 2 or 3 females, and perhaps their egg sacs and offspring. As you can see, basically, this is to give you a sense of how they are diverse in their morphology. They belong to different spider families, but I will show you that their nests are have a common structure in terms of, for the most part, being 2-dimensional and being these irregular structures. Here we have, this is the one exception to the 3-dimensional structure, species in the genus Ebutina, family Dictinidae, which is a really fascinating species because unlike the other species where the colonies grow to relatively large sizes before they produce daughter colonies, in this species, these colonies reproduce by fission, and the spiders also have a nomadic phase in their life cycle. And finally, this other really interesting species in the family of Oxyopidae, which you may know is a family of webless wandering spiders, but it turns out that there is a genus, the genus Tapinellus, where they have built or evolved these irregular 3-dimensional webs, and it is in that genus that a social species has originated. They are phylogenetically diverse. They happen in all continents other than Antarctica, but they have very similar social behavior. So in all of these species, they build some sort of communal nest. They cooperate in building and maintaining that nest. They have cooperative prey capture. They share their food, and they have communal brood care. We see here a hugely magnified head of a species in the genus Stegorhynchus that is feeding these spiderlings by regurgitation, and this may or may not be her own offspring. One of the things that makes the social spiders particularly interesting is actually the ways in which they are unlike other social organisms. Unlike the eusocial insects, for instance, they lack reproductive castes. So here we see a colony that contains multiple females with their egg sacs. So multiple individuals are capable of reproducing within colonies, both males and females, and they do so by mating with each other within the colonies. And this is the other aspect that differentiates them, not just from the eusocial insects, but from the vast majority of social organisms, and for that matter, most organisms. And it is that males and females remain within the natal nest to mate. And the consequence of that is that they are highly inbred with female biased sex ratios. And again, this is a topic that I will not have time to talk about today, but I'll be happy to answer questions later, or perhaps meet you in one of the coffee rooms later on. The relevant for the talk today is the geographic distribution of the social species. So we see here in the dots around here, the species in the 5 spider families where the social species have originated. And we see that they are all concentrated around the world's tropics. And within the tropics, if we go now to America, and we have here a magnified version of Ecuador, we see that they have a distinct distribution. The social species shown in red are only found in the tropical regions, and within those regions only at the lower elevations. We have the Andes going through Ecuador here, and we see that there are no red dots up there. There are no social species either at the higher latitudes. Instead, what we have in those regions are the subsocial species here shown in blue, which on the other hand are absent from the lowland tropical rainforest. So there are these two patterns. Subsocial species are absent from the lowland tropical rainforest, and the social species are absent from higher elevations and latitudes. And this is a pattern that I like to explain using the concept of environmental filtering. The other thing that we notice when we visit the areas where multiple species in the genus Anelosimus are found is that no two species seem to be alike. So for instance, here in this area in Ecuador, where only social species are present, we see that they have vastly different body sizes, from a species that is about 2 millimeters in length, another one is 5 to 6 millimeters in length. And in other areas where you can have multiple social systems represented, we see again that no two species have the same level of sociality. So these patterns suggest two questions. What explains the distinct geographic distribution of the social and subsocial species in the genus Anelosimus, which in terms of community ecology would be a question of whether there is and where are the environmental filters that determine this distribution. And the second question is whether those species within communities that seem to be different from each other, are they more dissimilar than expected by chance? So is there niche partitioning and limiting similarity responsible for the types of species that are present within local communities? So this brings me to the second 2/3 of this talk. And before I go on with that, I'd like to emphasize that this is a team effort, work that I have done over the years with students, undergraduates, grads, and postdocs at the University of Arizona, and more recently at the University of British Columbia. And I would also like to thank my advisor in Ecuador when I was an undergraduate close to 4 decades ago, Dr. Chiri Debris, who introduced me to social spiders, and he has also been instrumental in facilitating our work in Ecuador, and also co-advising undergraduate students from the Pontificia Universidad Católica del Ecuador, who have worked in our projects and are often co-authors in our papers. So let's deal with the first question. What explains the distinct geographic distribution of Anelosimus social systems? Is there evidence for an environmental filter? When we talk about an environmental filter, it's always going to be an interaction between extrinsic environmental factors and intrinsic organismal features. And I will argue that the key feature that explains why sociality has evolved in certain spider genera and families is the type of webs that they build, which are these dense 3D webs. And here is some drawings of the webs of the social spiders in the various spider genera where these species are represented. And we see that there are all these meshes involving, for the most part, a lot of silk. And this type of webs has some characteristics that facilitate the evolution of social behavior. In contrast, for instance, with an orb web, for instance, these structures are shareable, whereas the orb web is designed to be built and utilized by a single individual. In these irregular webs, individuals can occupy a common space and cooperate in pre-capture and brood care. These webs also presumably are costlier to build and maintain than other webs that involve less silk. They also are not just pre-capture devices as are orb webs, but they offer also protection against predators, and they should be subject to scaling laws of 3D objects. So let me briefly address the issue of cost. This is a topic that we are still working on, but in a preliminary analysis, what we have found, looking at the web content of these different types of webs, orbs, simple tangles, and sheet and tangle webs, and the social spiders, for the most part, build the sheet and tangle webs, we see here the total web silk content of those webs as a function of the size of the spiders or the size of the colonies. We have a social species here represented. And we see that there is we have a log scale here. We see that there is 2 orders of magnitude difference in the amount of silk contained in the sheet and tangle webs compared to the OR webs or simple tangle webs. So at least in terms of the amount of content, this is consistent with these webs being costlier to at least build. If we look here at the webs of the social species, we see that they are a nice continuation of the webs of the solitary species that build sheet and tangle webs. If we then take the data from these social species and we divide the web content by the mass of individuals in the colonies, so we look here at the web content per unit mass, we see that the amount of silk that individual spiders would need to produce to build these webs would decrease as a function of colony size. So there is an economy of scale that I would argue is relevant for explaining why these species may benefit to being in groups, in particular in areas where their webs may need constant repair, as we will see in a minute. So going back to the geographic distribution of the social and subsocial spiders in the genus Anelosimus, again, subsocial species absent from the lowland rainforest, social species absent from higher elevations and latitudes. To try to understand what factors may be responsible for this pattern, grad student Jennifer Guevara developed niche models of the species in this genus, social and subsocial species. And she looked at a variety of factors: temperature, precipitation, the variability of those, seasonality, productivity. She had something like 12 to 15 variables that she looked at. And the ones that emerged as being the most important and common both for elevation and latitude are temperature and precipitation. And we see here in black lines the probability of occurrence of the social species increases as temperature and precipitation increases, whereas the likelihood that the subsocial species will be present in very warm and wet areas drops completely. So what does temperature and precipitation have to do with the spider sociality? I would argue that rain precipitation has a direct effect because of the types of webs that they build, which I suggested already or showed to you that they contain a lot of silk and therefore are likely to be very costly to build and maintain. And we have found that the strength of the rain, so this is not just how much rain you have over the year, but how strongly it rains during a period of time. So we are looking here at rain rate millimeters per hour. We see that the strength of the rain is much greater at elevations in the lowland tropical rainforest than the rain happening at the higher elevations, which suggests that by sharing their webs, if they need to repair them frequently as they are damaged by strong rains, the spiders would benefit by being in larger groups, as I showed you before. The other pattern that we noticed is that predation rate increases with elevation, again, indirectly influenced by precipitation and temperature. And this is work of my student, Luis Camacho, which shows here how predation rate per hour decreases with elevation. And this is predation on insect baits, live insect baits that he deployed on both sides of the Andes. And again, because of high rates of predation here in the low elevations, it would pay off for spiders to be in larger groups, because if the mother dies due to predation, there will be other individuals around to look after the surviving offspring. We refer to this as the predation precipitation hypothesis, which basically argues that intense rains and strong predation exclude subsocial Anelosimus species from the lowland tropical rainforest. And here I'm going to describe to you a test of the predation precipitation hypothesis. Is work of master student Kathleen Hoffman at UBC. And what we did here is we took nests of a subsocial species that naturally occurs at a mid-elevation area and transplanted dozens of those to the lowland rainforest. And as a control, we also transplanted those within their native habitat, with the prediction that the transplanted colonies to the lowland rainforest will do much more poorly than those that remain in the native habitat, and that the effect will be due to rain and predation. So to test the causal effect of these factors, we use some very sophisticated high-tech methods, a tarp to protect the webs from the rain, and some sort of plate with tanglefoot to prevent predators from getting into the colonies. And we had some 4 treatments. We had no predators, no rain, no predators only, no rain only, and then we had some untreated nests. So 3 treatments where the webs were protected and 1 untreated treatment. And these are the results. First, we see here, as predicted, the nests that remained at the higher native site did a lot better than those in the lower elevation transplanted to the lowlands. But also, and this is one of the interesting results, is that the nests at the higher elevation, there was no difference of the treatments at that elevation, which is consistent with the idea that rain and predators being milder in that area are not a problem for the spiders that naturally occur there. These are the subsocial species, the mother with the offspring. However, these nests, mother with offspring transplanted to the lowland rainforest, did very poorly, especially when they were left unprotected, whereas those protected nests did better, suggesting again at least a partial effect of predation and rain as being responsible for the lower survival of these nests, these colonies in the lowland tropical rainforest. And what these results basically tell us, and this was actually a surprise for me when I started working in this, is that solitary living for a species in the genus Anelosimus is unsustainable in the lowland tropical rainforest. And we can see this in a different way also. We have data here of 2 social Anelosimus species. These species do occasionally produce colonies that contain a single female or just a few individuals. And we can look at the reproductive success of individuals in colonies of different size classes or different sizes. And what we see here is that those colonies that contain just 1 or a few individuals in both the species are below replacement value. So this would be the line where fitness would be 1. If populations of these species were to consist entirely of solitary individuals, they simply couldn't be in the lowland tropical rainforest, which basically corresponds to the situation here where below the replacement line, survival and persistence and replacement in these habitats is not possible unless the species, individuals in the species live in groups and cooperate. And in this particular case, I have shown you that the reason for that is because of intense rains and high predation, given the dense, costly webs that these spiders produce would not allow them to keep up with maintenance of the web and protecting the offspring. But interesting, when I first started working on this, I thought, oh, maybe they are social because there are all these big insects in the lowland rainforest that they could only access if they cooperate. But it turns out that that is not the reason why they are social, at least not to begin with. The other pattern that we need to explain is why social species are absent from higher elevations and latitudes. So for this, we go back again to our precipitation and temperature patterns. And what we have found is that these have an effect on the size of the insects, which we see here that insects at the lower elevation, so we are looking here at insect size, both for, here's diurnal insects in black, the nocturnal insects, and we see that those at the low elevations are considerably bigger than those at the higher elevations. And in a second study, we show here how that is reflected in the distribution of insect sizes. And what's happening is that at the higher elevations, the larger insect size classes are for the most part missing, whereas they are plentiful at the low elevation tropical rainforest site. And just to give you a sense of scale here, we have a log scale here. So these are quite significant differences in the size of the insects. And these brought us to develop what we refer to as the insect size hypothesis, which basically suggests that absence of large insects at the higher elevations, so in these areas, excludes social species from occupying these areas, so higher elevations or also higher latitudes. And the reason for that, and I will show you in a minute why that is is because of three-dimensional scaling of the webs that they build. So what does insect size matter, and what does this three-dimensional scaling have to do with it? As you know, as three-dimensional objects grow in size, the surface area doesn't grow as fast as the volume. So if we were to divide surface area to volume ratio and plot that as a function of the volume, here both of these in the log scale, we see that that ratio would be a decreasing function of the size of the object, in this case, the social spider colonies. And the reason why that matters is because the number of insects that are intercepted by the colonies as food for the spiders is proportional to the surface that the webs are presenting to the environment, whereas the amount of food that is needed is proportional to the volume, which is proportional to the number of spiders that are occupying those webs. So a prediction that we can make from these geometric considerations is that the number of prey per capita should be a decreasing function of colony size. We test this hypothesis in both a subsocial species, so again here mother with her offspring, the offspring together for a while, at a high elevation cloud forest area, and in a social species in the lowland rainforest. And we see that in both cases, number of insects per capita decreases with colony size. And just to gain a sense of scale here, we have log scale for both of these here. So there are pretty significant differences in the size of the colonies here. The pattern that we observe for the species in the rainforest is quite interesting. Why would these spiders be in these really huge groups if they are getting fewer prey per capita? Well, it turns out that because they are in an area where large insects are abundant, they are able to cooperatively capture increasingly larger insects as colony size increases. And if you put together fewer insects times bigger insects, what you get is biomass per capita being maximum at intermediate colony sizes. The shape of this biomass per capita function would explain both why they are in groups. They do better in groups of intermediate size than in smaller groups, but also it would explain why eventually they disperse. If we go to the higher elevation area, on the other hand, where these large insect size classes are missing, the spiders may be trying to cooperate to capture bigger insects, but the bigger insects just simply aren't there. And as a result of that biomass per capita simply decreases as colony size and also the size of the individuals within those colonies increases. And this would explain why the subsocial species in these areas disperse before producing a new generation of offspring at the same location. And this brings me again to these graphs here. Why and where are they social? I show you that the basic reason why they are social is because if they weren't in groups, given the types of webs that they make, they couldn't be in areas where rain and predation are strong. But it turns out that insect size, that was my first intuition, does matter, but it's more a requirement than a reason why they are social to begin with. And in order to be social, large insects need to be present in the environment, which fortunately for the spiders and for us studying them, large insect size coincides with high predation and high precipitation because of the way temperature and precipitation correlate with these factors. So relating this again back to these questions in community ecology, what are the environmental filters of the spider sociality in the genus Anelosimus? I have shown you that it is a combination of insect size with the small insects at the higher elevations, excluding the social species from being present in those areas, whereas the large insects at the lower elevations allow them to be there. And on the other hand, the subsocial species are excluded from the lowland tropical rainforest by strong rain and predation. But they do just fine at the higher elevations where these factors are mild. And with that, I'll move to the second question that relates these issues to community ecology. And that is, what determines the composition of local communities in terms of the ecological niche that the different species occupy within those areas? So is there niche partitioning and limiting similarity? And to get into that, well, this is the last part of the talk today, sociality, niche partitioning, and limiting similarity. So a brief review of niche theory. It has been understood for a long time that the species within an area tend to be different in specific ways that allow them to use resources differentially. And studies have typically focused on things like body size and morphology as being responsible for differentiation of the species. For instance, we have an example here of Darwin's finches, how the size of their beaks corresponds to them using different sized and seeds with different hardness, or these lizards in the Caribbean that differ in body size, the ratio of the legs to the body, and other factors that determine their use of space and resources. And my argument today is that differences in group size and level of sociality may also lead to differential resource use and play a role in community assembly. And going back again to this figure that we saw before, this is a representation of various communities where multiple Anelosimus species are represented. And we see that within these communities, no two species seem alike. So we can ask a few questions about these communities. Are these differences associated with differential resource use? And are species within communities more dissimilar than expected by chance? And addressing both of these questions would be tests of niche partitioning and limiting similarity. So taking on the first question, are these differences associated with differential resource use? We go to this area in Brazil, Sao Paulo, Serra do Japi, where species with different levels of sociality are represented. And for work in this area, I'd like to thank my collaborator, João Vasconcelos Neto, and his students and postdocs who were instrumental in the work that we did in this area. Serra de Japi is a very special place because it contains this broad range of social systems in the genus Anelosimus. There is a solitary species. There are two subsocial species. One is relatively uncommon. And then there is this intermediate social subsocial species, which builds relatively large nests, but within these nests, the spiders tend to be spread out, occupying what appear to be like individual territories, and a fraction of individuals disperses every generation, but some others remain behind. But they don't seem to be as cooperative as are individuals within the more social species, where everybody's mixed within a common space and they seem to cooperate in pre-capture and also in brood care. So the question is whether these differences in level of sociality are associated with differential resource use. And they are indeed in pretty extraordinary ways, actually. First, we see here how they differentiate in their use of space. The three less social species are more common towards the forest edge. We have a log scale here on distance from the forest edge. With their smaller nests, they tend to be more likely to be present towards the forest edge, whereas the social species with larger nests are found more towards the interior of the forest. Within these two microhabitats, they differentiate in the height from the ground at which they happen, and also on the position on the branch, the core, the middle, or the tip. As if this wasn't enough, they also seem to be differentiated in their phenology, with the species being in slightly different parts of their life cycle at a given time of the year. For instance, in the two more social species are offset by a period of a month in the peak of the reproductive period. They are also differentiated in the use of resources. In this case, we see in the size of the prey that they capture. We see that the least social species captures the smallest insects. The subsocial here is insects somewhat bigger. The intermediate subsocial bigger insects, and the more social species even bigger insects. And these differences are significantly different. And as I mentioned before, there was a difference in how cooperative these two species are here. I will not have time to show you the data, but let me just sort of point out that the difference in the size of the insects that these species are capturing is not really a difference in the size of the colonies, which seem to be fairly comparable, but rather in the extent to which individuals within the species are involved in cooperative prey capture. These individuals in these intermediate social subsocial species are not very cooperative, capture prey individually for the most part, whereas these other ones are highly cooperative, and that creates the difference in the size of the insects that they capture. So yes, there are differences associated with a level of sociality in terms of the resource use of these spiders. What about body size? For this, we go to this area in Ecuador where there are these vastly species, all social, with vastly different body sizes. And we're going to look here at the largest species and the intermediate sized species. They are more or less to scale here to give you a sense of how different they are in size. But they build very similar webs in their architecture, and they can happen sympatrically nearby each other within a few meters of each other. We look at the insects that they are capturing. We are seeing here the total biomass, and we see that the smaller species capture insects in the small size range, and the larger one captures insects in the whole range, but mostly it's getting prey from the larger size classes. The small species here would seem to be a bit in trouble because it has an included niche within that of the larger species, but it turns out that the smaller species is more efficient at capturing these small insects and therefore creating niche differentiation between the two. So yes, there are differences associated with body size in terms of the use of resources of these spiders. And the last question that I would like to address is whether species within communities are more dissimilar than expected by chance. So is this pattern that we observe just a coincidence, or is what we are seeing here something that cannot be explained just by chance? And to address this question, we looked at 53 communities across the Americas that have varying compositions in body size and level of sociality. This is where these communities are located. They contain anywhere from 2 to 5 species. And what we did is we obtained a functional dispersion index, how spread out within these communities they were in terms of body size and level of sociality. We obtained the observed functional dispersion index, and then we obtained the index that we would see if these species have been assembled randomly with respect to body size and level of sociality, taking into account that there is these environmental filters in terms of where social and subsocial species can occur. And this is the distribution of that index that would be expected if these communities were randomly assembled with respect to body size and level of sociality. And the value that we observe, as you see here, is significantly different from the expected. So yes, we can show that the species within communities are more dissimilar than expected by chance. So just to conclude, I have shown you that there are environmental filters, insect size, predation, and precipitation intensity that would explain where social and subsocial species in the genus Anelosimus are found. And there is also niche partitioning and limiting similarity that would explain what particular species happen together within local areas. And what I would like to suggest is that these lessons that we have learned from studying the biology of social spiders can be extended to understand other social systems and also other levels of organization, both in terms of where the social and non-social species in different groups are found, but also what particular species happen or do not happen together within ecological communities. And with that, I would like to thank you for your attention. I would like to thank my funding sources, sponsorship of the institutions in Ecuador and Brazil that helped us and made this work possible, field station and native lands, and the wonderful people that live in these areas that have helped them maintain in beautiful condition, which we hope we will be able to uphold for generations to come. And with that, I would like to take any questions.